Hello everyone. Uh, this presentation was combined by the bioinformatic member of Landmark University team led by Professor Marion Adebiyi, and the presentation is exploring ovarian cancer subtypes and prognosis prediction using PSO with CNN. Ovarian cancer is a type of cancer that begins in the ovaries, where the eggs are produced in the female reproductive system, and it can also start in the fallopian tube. Ovarian cancer is often diagnosed at a later stage when it is more difficult to treat. Years back, it is used to be more common in women over 50, but more recently, it is seen in women in their 30s and 20s. Most time, treatment involves surgery to remove the tumor, followed by chemo. The multiplicity of ovarian cancer contributes to its complexity. It has several subtypes, each with distinct molecular and clinical characteristics. The symptoms are often vague and can include abdominal and pelvic pain. Here are just about 7 of the subtypes with their symptoms similar. Their symptoms are quite similar. Because of our time, we would just like to move on. And for omics data, it captures different disease feature, and that calls for a model that can analyze such variation of data. Understanding the distinction is crucial for targeted therapies and better patient outcomes. For this study, we employed advanced analytical techniques like particle swarm optimization combined with CNN to help us uncover those hidden patterns in the multi-omics datasets. Here is the methodology. Here we made use of 7 ovarian cancer datasets. That's the title of the dataset that is publicly accessible from Cargo repository. The dataset contains gene expression profile, clinical data, DNA mutation information, genetic alterations, and subtypes of ovarian specific characteristics. Moving on to the data pre-processing. As we all know, CNN is often used for images, but just for the novelty of this job, the novelty of this study, where a lot of pre-processing were done just for us to make use of CNN model. The DNA sequence were passed with their corresponding classes. The sequences were tokenized at character level into numerical tokens, and the numerical tokens were then padded to ensure uniformity across samples. And finally, the class labels were encoded into numerical format for further pre-processing and analyzing using our particle swarm optimization. Particle swarm optimization helped to analyze and gave us an optimal parameter which we use CNN to which help us to train our CNN model. The data was split into training and testing. The input data was reshaped to fit in our CNN model. And for the first layer, which is convolutional layer, it extracted the features needed. The features were then sampled by the mass pooling layer just to help reduce the dimensionality. And of course, the dropout layer was included for regularization before the two-dimensional outputs were flattened into one-dimensional vector, and before the fully connected layer classification. On the other side of this slide is the step involved in the data pre-processing. And for results, as you can see, we have our images, our visualization showing how well the model trained, and our validation accuracy. The model achieved a validation accuracy of approximately 97.22%, and these metrics include a high effective model capable of accurately identifying molecular features and correlating them with clinical outcomes. In conclusion, this study demonstrates the effectiveness of PSO CNN in analyzing multi-omics data. Here are references and acknowledgement. We would like to appreciate the body ISCB for ISMB 2014 and our research group in Landmark, in our great university Landmark University, for this opportunity. Thank you.